Hello students, once again welcome back to biology class. Myself Dr. Raghavendra here. Well, in the earlier class, yes, we are discussing about the biomolecules. In that we discussed about the concept as such, dynamics of the metabolism. So metabolism as such we consider it is the sum of or the total of anabolic and catabolic reactions, definitely. So as such, now we also discussed about metabolism is the basis of living also. Then what is the state of living? Let us understand that. Further we will discuss about the enzymes also. When we talk about the living state or the living state means in what circumstances we call the body as a living body, not the dead body. In what circumstances we call the living as it is a living body, not a dead body. Why? Because so there is a life, so life exists in that body, that's why we say it is a living. So as such, what do you mean by living? Living refers to, yes, living is active state of metabolism. You can say living refers to active state of metabolism that exhibit certain basic characteristic as such the growth, maybe respiration, maybe reproduction. Okay, so there is what you can say mainly responses to the stimuli. Okay, consciousness we say. So all these are the basic characteristic of the living. Growth, so cell division or cell repair, reproduction, okay, consciousness, all these are the what we can say mainly the respiration, all these are the characteristic features of the living. So the living organisms have millions of cells, and definitely what we can say that mainly the living cell exhibit thousands of biochemical reactions or we can call them as the biochemical pathways we call them. Yes, so these pathways, biochemical pathways, can be linear, straight chain, yes, keep on happening. It can be glycolysis, it can be electron transport system, ETS we can say, transport chain we can say. Okay, these are linear, cyclic pathways. They can be cyclic. It can be Krebs cycle. It can be what you can say one more important ornithine cycle, many things. So biochemical pathways are either linear or they may be cyclic pathways, definitely. So as such, metabolism refers to what you can say. Metabolism is again, we need to remember that it is the sum of, metabolism is the sum of, yes, as such, catabolism and then anabolism. So catabolism example, let me mention one more. Starch broken and forming the glucose plus glucose molecules. Okay, this is what we can say cutting up of mechanism or breaking of mechanism. Anabolism, again simple example. So mainly we can say that other than that, yes, glucose molecules can be broken into, further the glucose can be broken into lactic acid. This is also, or glucose molecules can be formed in the pyruvic acid. These are what we can say catabolic reactions. So when we say anabolism, mainly example for anabolism, yes, glucose plus glucose forming the glycogen It is one anabolic reaction. Other than that, photosynthesis. Photosynthesis also one of the anabolic reaction. Well, so when you are talking about the living state as such, what is the basic definition when we talk about the living state? So, living state is living state is non-equilibrium steady state. It is a non-equilibrium steady state to be able to perform the work or functions. So, why this statement is given? We need to think mainly to tell you living state is non-equilibrium state. Yes, it is acceptable. Why? Because let me tell you when we say non-equilibrium, because in a living cell, all biomolecules cannot reach same level of concentration. If it happens, the cell may die. Example, glucose, it is 4.5 to 5.0 millimoles, whereas hormones concentration can be nanograms per ml. So hormones are very least, very small concentration, whereas the glucose level is much more higher. That means all the biomolecules cannot come to a equal state or equal concentration. If all the biomolecules reach a equal concentration, cell will die. Okay, so that's what even in the physics it is stated. A system at its equilibrium or static state, it becomes non-functional. Okay, so thus we can say that in physics, according to physics, system at equilibrium is non-functional. So system at equilibrium is non-functional, it is static, it is not doing any work. But all these biomolecules maintain their own different level of concentration, separate level of concentration. So there is a non-equilibrium always happens, and every cell tend to remain as a non-equilibrium only, not as a equilibrium. If it go to equilibrium, it becomes the non-functional. So only they have to maintain, yes, non-equilibrium state. So only in a living cell, all biomolecules cannot reach the same level of concentration. So mainly the glucose can be measured in terms of molar values or millimoles, whereas the hormones level can be measured in the nanogram. That means each biomolecule has their own level of concentration or optimum concentration that make the cell to remain active and to perform its function. Otherwise, the cell will die. That's why it is stated living as well as metabolism are synonyms. So you need to remember this mainly. Living and metabolism are one are the same, synonyms. Okay, so here you need to remember living and metabolism are the synonyms. This is all about what we call as living state. What do you mean by living state? It is the active condition or active state of metabolism. So in a living cell, it exhibits thousands of biochemical reactions And these biochemical reactions or biochemical pathways, they may be linear, they may be cyclic. So glycolysis, electron transport chain, these are all the straight, what you can say, the chain reactions or the linear, whereas cyclic pathways, Krebs cycle as well as the Ornithine cycle are the cyclic pathways. Their metabolites are again reutilized. Okay, so that's why they are the cyclic pathways. Whereas when I talk about so metabolism is overall catabolism as well as anabolism. These keep on happening in a living cell. So starch is broken into glucose molecules. Glucose molecules in the presence of oxygen is broken into two pyruvic acid molecule, that is called glycolysis. Or glucose molecule in the absence of oxygen in the muscle cells, it will be converted into lactic acid, anaerobic respiration. So thus braking mechanism keep on happening to yield the energy. Energy is in the form of ATP again. So whatever the biochemical activity happens inside our body, that gives us the strength. So only we are able to do the lots of physical activities. It may be the running, it may be the jumping, it may be the walking. So all these physical activities happens when there is a biochemical activities are happening perfectly inside our body. Then only there is the energy produced. Now catabolism is mainly energy yielding or energy releasing. Anabolism is energy consuming reaction. So glucose plus glucose become the glycogen. Photosynthesis itself, where the what you can say carbon dioxide as well as the water are mainly used, and converting them into the what we mainly the glucose as well as the release of the oxygen. The photosynthesis also best example of anabolism only. Photosynthesis also best example of anabolism. So basic definition: living state is a non-equilibrium steady state. So and to be able to perform the work or functions. If what happens if it is not in a non-equilibrium? Yes, all the biomolecules level reach to the same line, so they become equal. Okay, whether the protein, carbohydrate or lipid, or everything become equal. Then what happens here? System at equilibrium become non-functional. The cell may die. Okay, so in a living cell, all biomolecules cannot reach the equilibrium state. So or same level of concentration cannot possible. So thus there is a different concentration for each of these biomolecules, or we call them as the optimum concentration, that helps the cell to survive, to repair itself, to grow, to multiply. So thus living and metabolism means a cell have to live or a organism have to live. Metabolism have to happen inside the body. So that's what we call it. Living and metabolism are synonymous. They are always one and the same. So this is what we have discussed all about mainly the living state. So we need to go further regarding what we call as the next concept, all about enzymes we will be discussing. So here let us go ahead with the next concept. Hope you have noted down these points. Let me just go ahead with the next concept, what it is called as regarding the enzymes. Fine. So since from the school days you people are studying enzymes, hope you may be knowing the basic definition of enzymes. What are the enzymes? Enzymes are basically, yes, basically they are chemical nature. What? Are they nucleic acids or the lipids or the carbohydrates? They are not the nucleic acids. Yes, they may be few. Yes, nucleic acids do exist as enzymes. They may be, but they are not carbohydrates. They are not lipids. They are perfectly the proteins. Fine. So the next concept under the biomolecule we are going to study all about enzymes. So enzymes is the most important concept under this chapter. So let us study about the enzymes. So enzymes, the term coined by Kuhne. So the scientist who gave this term, and under this enzyme we will be discussing about properties of enzymes. Second, rate of reaction, and most important examples we discuss: activation energy, Number 4, we will be discussing about nature of enzyme activity. Number 5, factors affecting enzyme activities. Number 6, classification of enzymes. We will discuss. Number 7, we will know about some more important information such as cofactors. Okay, well, so enzymes are basically, by definition, we can say that these are the biocatalysts which catalyze various kind of biochemical reactions. So let us understand enzymes are biocatalysts catalyze or accelerates the rate of biochemical reactions. Okay, so thus the enzymes are very essential in each and every kind of biochemical reaction. So enzymes are basically biocatalysts as such. By nature, what they are? Enzymes by nature. So chemically enzymes are mainly proteins. Yes, chemically enzymes are proteins. Okay, and yes, they are made up of amino acids. So like other proteins, enzymes also exhibit. So enzymes exhibit primary, secondary and tertiary structures. In tertiary structure, they have crisscross, okay, linking loops. They have crisscross linkings, loops, and what we can say that crevices or pockets or folds that forms the active site. What is the use of active site we will discuss later. Okay, so thus enzymes are biocatalysts, then they are mainly or basically by nature they are the proteins, and these enzymes are basically made up of long chain of amino acids. The long chain of amino acids, when they keep on folding, making a loop of themselves, they create a kind of pocket like structure or crevice like structure. Crisscross linking will be there. So with that, yes, they form the active site, and active site is very important for binding with the substrate. So at the time of biochemical reaction, yes, the enzyme need to bind with the substrate. The enzyme need to break down or convert whatever that substrate into product. That is, mainly the basic nature or the properties of enzymes. So one more important thing as such, enzymes can be, so enzymes can be nucleic acids, or yes, nucleic acids can be acting as enzymes and are called as, and they are called as what? They are called as ribozymes. Ribozymes. So other important property. So here what we have discussed so far, number one, we discussed the enzymes are biocatalysts and they catalyze the biochemical reaction. Chemically they are proteins and they are made up of amino acids. Okay. So enzymes exhibit the third property, yes, primary, secondary, tertiary structure. And the fourth property in tertiary structure, they have the loops or the pocket like structures or crevices and that form the active site. And fifth point, enzyme can be nucleic acid, yes. So they also called as ribozymes. And the sixth important property, enzymes are heat and pH sensitive. Beyond the optimum range of heat, beyond the optimum range of the pH, enzymes may become denatured or inactive. So beyond the tolerable range, so they become, being a proteins, they become denatured and inactive. Okay, so this is the most important property again. Number 7, enzymes as such, we can say that the enzymes are heat sensitive. Yes, enzymes activities with change in every 10 degree, change in every 10 degrees centigrade. Okay, so the enzyme activities with a change in every 10 degrees centigrade, it may be doubled or reduced to half. Means the efficiency of the enzyme, rate of reaction catalyzed by this enzyme, if 10 degrees centigrade may be increasing more, yes, there may be, the rate of reaction also may increase. Some of the enzymes will be acting fast in the increase with the temperature, but some of the enzymes which are heat sensitive, they will definitely fail over there, they may become denatured over there and they will not perform any function, or the rate of reaction may be reduced to the half. This is very important point again with the properties of enzymes. Number 8, one more important property. Yes, there are certain exceptional cases where we can say that the enzymes may be resisting temperature fluctuation. So the enzymes can be thermostable. Enzymes can be thermostable, found in thermophilic bacteria, so such as the enzyme Taq polymerase, Thermus aquaticus we say, Taq polymerase, which is used in the PCR reaction. The enzyme Taq polymerase remains active, okay, so beyond 80 to 100 degrees centigrade, it is found in a bacteria, Thermus aquaticus, found in bacteria, Thermus aquaticus. Thermus aquaticus, yes, seen in hot springs, hot spring, and thermal vents, thermal vents. Let me tell you about this. I hope you will be able to see these points clearly. So here what we are observing basically are the important or basic properties of enzymes. Enzymes are biocatalysts. Yes, their function is mainly to catalyze the biochemical reaction. Whether it may be the cyclic or linear pathway, they will catalyze. Chemically, enzymes are proteins and basically they are made up of amino acids. Amino acids are attached with one another mainly by the, what we can say, that peptide bond. And then these amino acids, yes, we call them as mainly, they form a long chain polypeptide chain. Fine. And then, so enzymes mainly exhibit primary, secondary and tertiary structure. When they become tertiary structure, they have lots of loops, they have lots of long chain, they have pocket like structure or crevice like structure that form the active site for the binding with the substrate. And then, so enzymes can be nucleic acids, yes, they are also called as ribozymes. Enzymes are the heat and pH sensitive. They have their own optimum range of pH, optimum range of temperature. Beyond that, enzymes will become inactive, denatured. They will lose their efficiency. Enzyme structure is altered. It cannot bind with the substrate. Fine. Enzyme activities with the change in every 10 degrees centigrade. Yes, enzyme activity may go high. It may become double efficient or it may become very much inert or very much weak. Then half of its reaction rate will fall down. That is the thing. Enzymes can be thermostable. Yes, certain enzymes which are isolated from the bacteria, and the bacteria, they are mainly thermophilic. They have the love for the heat. So they are found in the sulfur vents, we call them. We call them as hot springs. Wherever the water source is very much close to the volcanic eruptions, there we can find volcanic eruption also there, water also there. Eruptions can be seen even inside the ocean also. And in such condition, water will be boiling, and such boiling water, there also bacteria survive. We call them as a thermophilus bacteria or Thermus aquaticus bacteria. From such bacteria, a enzyme is isolated called as a Taq polymerase, which can remain very active even at a temperature of 80 degree to 100 degree. So most of the metabolic activities, most of the enzymes will become denatured at a 40 degree temperature itself. But this enzyme will remain highly active, highly efficient, which is isolated from the bacteria Thermus aquaticus, which is seen in hot thermal springs or thermal vents, we call it as. So this is all about what we say, the basic properties of mainly enzymes. The rate of reaction. So when we talk about the rate of reaction, there are variety of reactions. Physical reactions, chemical reactions. Chemical reactions are organic reactions, inorganic reactions. And then these reactions can be catalyzed reaction, uncatalyzed reactions. So rate of reaction again it depends on amount of product is produced per unit time also. Yes, we will be discussing with all these variety of reactions or rate of reaction, and next concept as such the activation energy, and then the mainly the nature and enzyme activity also we will be discussing, how actually the binding of substrate and enzyme, how this enzyme convert that mainly the substrate into product, the basic structure of enzyme, what do you mean by apoenzyme, what do you mean by holoenzyme, all that we will discuss in the next class. Okay, till then take care, study well. Thank you very much.